Welcome students to our online classes. Myself Ashwini Honkarse is going to explain you the lesson the fundamental unit of life part 2. So in part 1 we studied index of the lesson. Yes. Then afterwards we studied introduction of cell life, unit of a life, that is cell, how the cell look like, who discovered the cell, how the discovery of journey of cell is there with the help of microscope. Then we studied about different scientists who worked on cell as well as cell theory, right? Then we studied types of organisms, in that unicellular and multicellular. Now we are going to study about types of cells. So before study the types of cells, let us take a journey about cell, how it's look like. Yes, how it look like. So let us see how the cell is look like. So students, now just observe, this is a type of cell and it is called as a eukaryotic cell. So what is eukaryotic cell, that we will see. This is a model of eukaryotic cell found in humans, animals, plants and fungi. Okay, so this is a one eukaryotic cell. Now just see, this is a rough endoplasmic reticulum which is lavender in color. This is rough endoplasmic reticulum. Manufacture and packages protein. It is a manufacturer and packages protein. Now see the nucleus. So this yellow color outer bounding circle is called as a nucleus. The nucleus contains the cells and genetic material, that is called as a DNA. Now see, this is a liquid jelly, that is called as a cytoplasm. You can see a water-like structure over here. This is called as a cytoplasm. The cytoplasm is a semi-fluid gelatin-like a substance that surrounds the organelle inside the cell. Now see, the thread-like structure, these are called as a chromosomes and we can call it them as a DNA strands. These called as a DNA strands. Now see, the nucleus surrounded by the yellow color structure, this yellow color line is called as a nuclear membrane. This is called as a nuclear membrane. Now these pipe-like structures, they are called as a centrioles. These are called as a centrioles. The centrioles play a crucial role in cell division and the location of nucleus. So what they decide? The cell division and the location of nucleus. Now see, this is the cytoplasm. This is called as cytoplasm. A cytoplasm is a semi-fluid gelatin-like a substance that surrounds the organelles inside the cell. So this all is called as a cytoplasm. So again these are chromosomes, nucleus. Then see that these are called as a nuclear pores. You can see the yellow color outer structure. These are nuclear pores, act as a doorways between the nucleus and the cytoplasm. So this is the semi-liquid cytoplasm and this is the nuclear boundary and these are called as a doorway. So from these blocks the food and other materials will exchange from cytoplasm to nucleus. Now these green color, they are called as a mitochondria. So you now understood this is a plant cell. This is what? A plant cell. So mitochondria act as a power plant of the cell. It is called as a power plant of the cell. Now see, again this is a cytoplasm, chromosomes, mitochondria. Then this outer boundary is called as a cell membrane. It is called as a cell membrane. So, cell membrane encloses and protects the content of the cell. So, cell membrane is present so that all materials will be protected inside the cell. Now, you can see the ribosomes here. This is called as a ribosome. Ribosomes read instructions from the messenger RNA to synthesize protein. So, RNA give the message to the ribosomes to synthesize the protein. So, this is called as a ribosomes. So again this is a nucleus. This is called as a nucleus. So these are again nuclear pores. This is a nuclear membrane, cytoplasm. So I hope you understood. Now these are vesicles. They are called as a vesicles. Vesicles stores and transport substance within the cell. So what is the duty? They will store and transport the substances in the cell. Okay, so now you observe the full structure of cell, right? Now this red color structure is called as a Golgi apparatus. They are called as a Golgi apparatus. The Golgi apparatus modifies as protein and sends them to other parts of the cell. They will be modified proteins. So they are modified proteins and they may send them to other parts of the cell. Now these yellow color pipe, they are called as a smooth endoplasmic reticulum. Just now we studied endoplasmic reticulum. That lavender color, it is a hard endoplasmic reticulum, and these yellow color, they are called as a smooth endoplasmic reticulum. The smooth endoplasmic reticulum synthesizes lipids, then phospholipids and steroids. Now you can see a thread like a structures. They are called as a cytoskeleton. They are called as a cytoskeleton. The cytoskeleton gives support and structure to the cell. It gives support and structure to the cell. So students, I hope you understood how the structure of nucleus cell is present and which type of organelles are present in the cell. So let us study now the types of cell, that is prokaryotes and eukaryotes. What is the difference between them? Cells of organisms lacks nuclear membrane. So in a prokaryotic cell, the nuclear membrane is absent. Nuclear membrane is absent, that is yellow color outer boundary is absent. Cell of organisms have a nuclear membrane. Here nucleus is absent. Here nucleolus is absent. Here nucleolus is present. So nucleus contain another structure inside it, that is with DNA, that is called as a nucleolus. It is absent in prokaryotes and it is present in eukaryotes. So it contains single chromosomes, that is DNA. It contains maybe single or multi type of chromosomes. So in prokaryotes, reproduction is always asexual. So you already know what is sexual and asexual reproduction. So here eukaryotic reproduction is both sexual and asexual. So in prokaryote always it is unicellular. It is always unicellular. Often it is unicellular and it may be a multicellular also. Membrane bound cell organelles are absent. So membrane bound cell organelles are absent. They are floating in nuclear in the cytoplasm as how anyhow. The membrane bound organelles are present like a mitochondria. So different types of organelles are present like mitochondria, Golgi bodies with their membrane. Then here centrioles is absent. Here centriole is present only in animal cell. It is not present in other cell. Centriole is present in animal cell. So cell division is by binary fission. So what is binary fission? What is mitosis, meiosis? That all we are going to study. Here cell division is mitosis or meiosis. For example, bacteria, blue-green algae. So these all contain a cell, that is prokaryotic cells, and we already know that they are single cellular organisms. Here the example is fungi, plant cell, animal cell, etc. Now you can see these are the diagrams of animal cell and plant cell. So please draw these diagrams neatly and just see as how we studied the structure of cell. Only little bit difference is there between plant cell and animal cell. So let us study what is the difference between these two. So here the animal cell and here plant cell. You just observe the structure and afterwards you study the cell difference. The cell wall is absent. So where is the cell wall? Here the cell wall is absent. So here the cell wall is present. You can see the cell wall over here, right? The inner side layer of the cell. Now next, here cell wall is present. Then plastids are absent, here plastids are present. So you can see the plastids over here, they are present in animal cell but not in plant cell. Now see the next one. Here in animal cell, the centriole are present and here the centrioles are absent. Here Golgi bodies are present. Golgi bodies are present and called as a dictosomes. Then vacuoles are absent. If present, they are very small. Here vacuole are present and in large in size. Then centrosome is present with one or two centrioles. Here centrosomes is absent. So you can observe in the structure of plant cell and animal cell. So what are those and where they are present? You just observe by the diagram. Now we are going to move towards the process of diffusion. So what is diffusion? Now see, the spontaneous movement of a substance from the region of high concentration to the region of low concentration is called as a diffusion. So for example, if amoeba is present, and what the amoeba is doing, when it will take the food, it will digest the food and it will respirate also. That time it will take oxygen and leave carbon dioxide. But when it is taking oxygen and when leaving carbon dioxide, at the time of diffusion, the process of diffusion is going on, because when it will take oxygen, the inside body concentration of oxygen is very less, so it will take the oxygen inside. When the carbon dioxide concentration will be increased, that time it will leave the carbon dioxide outside. Okay, some substances like carbon dioxide or oxygen can move across the cell membrane by the process of called diffusion. Cell also obtains nutrition from the environment. So by the process of diffusion only, the other cells, not only amoeba, the cells are getting nutrition in their body. Now see the process of osmosis. So osmosis, the word related to water. The word is related to water. So the movement of water molecules through selectively permeable membrane. Selectively permeable membrane. So what it means? So it means it will allow the substances to pass through it. They are selective. Only selective substances may be passed through that membrane. So that's why we are calling it as a selectively permeable membrane. Along the concentration gradient is called as a osmosis. So what is osmosis? The movement of water molecules through selectively permeable membrane along the concentration gradient is called as a osmosis. So what will happen? What exactly it mean? So sometimes what happen? Plant and animal cells are involved in the process of reproduction and digestion. So that time water molecules will be released, right? Or when they are preparing the food, they need also water, right? So when they need water, the water goes inside the cell, and when they don't want water, the water goes outside from the cell. When it need water, it will takes from the atmosphere. When it will be leave, that time we can call, at the morning time you can see the droplets, right? You can see the sweat from your body. So these all processes involved in the process of Osmosis. Plant cells tend to obtain water through osmosis. Now see what is the difference between osmosis and diffusion. It is limited only to the liquid medium. So osmosis, it is related to only the liquid medium. So diffusion occurs in all medium, solid, liquid and gases. So the membrane which allows by the concentration, high concentration to low concentration, it may be solid, liquid and gases. Osmosis requires a semi-permeable membrane, but it do not requires a semi-permeable membrane. Osmosis depends on the number of solute particles dissolved in the solvent. So it depends on number of solute particles dissolved in the solvent. It depends on the presence of other particles. What are the particles present? In on that the diffusion is depend. So it requires water for the movement of particles, and it do not require water for the further movement of the particles. Only the solvent molecules can diffuse, and both the molecules of solute and solvent can diffuse. Here the flow of particles occur only in the one direction, where in the osmosis. So when water will be don't want, that time it will be released. So it will be of one only. The flow of particles occur only in one direction. The flow of particles occur in all the direction. So now you can see the direction over here. So when it needs, the water is going inside. Now hypotonic, hypertonic and isotonic solution. What happened in a cell in sugar or salt solution? So now you can observe the structure of cell over here. Here the water concentration is more compared to the cell. Here water and the cell both are same, cell fluid is same. Here the water concentration is, then here water concentration is less compared to the vacuole. So here it is plasmolized, here it is flaccid, and here it is turgid. So what happened over here? So hypotonic solution means what? The medium surrounding cell has a higher water concentration than cell. So the medium of the surrounding is contain more water compared to the cell. So that is nothing but hypotonic solution. So results what? Cell will gain water by osmosis and likely to swell up. The next, isotonic solution. Medium has exactly same water concentration as the cell. Here both are same. So water crosses the cell membrane in both direction. Cell will stay the same size. Next, hypertonic solution. Medium has lower concentration of water than the cell. So water crosses the cell in both direction, but more water leaves the cell than enters it. So this is the result of these three: hypertonic, isotonic and hypotonic. Now next, so plasma membrane or cell membrane. So just now we studied about the cell. Let us study how the plasma membrane look like, or cell membrane, outer cell membrane look like. So it is made like this. You can see the structure over here. This is the outer line of the cell. This is outer line which we studied. Yes, now see. This is the outermost covering of the cell that separates the contents of the cell from the external environment. So because of this membrane is present, so internal part of the cell are safe. The plasma membrane allows or permit the entry and exit of the some materials in and out of the cell. So this membrane, what it will do? It will selectively item only it allows to be inside and goes outside from the cell. It also prevents movement of some other materials. The cell membrane is called selectively permeable membrane. So what we may call it as selectively permeable membrane. It is made up of lipid and protein. So this is very important. The cell membrane is made up with a lipid and protein. Now next is called as, so now let us study properties of plasma membrane. What are the properties? So it is flexible. It is what? Flexible. It is made up of organic molecules called lipids and proteins. That's why it is very flexible. Its flexibility enables cell to engulf in food and other from the external environment. This process is called as a endocytosis. So what is it process? It is its flexibility enables cell to engulf. Engulf means to take inside in food and other from the external environment. So amoeba acquire food through this process. So we already studied that how the amoeba is taking food. It enlarges its legs that are called as a pseudopodia, and that pseudopodia take the food and again it will comes to the normal structure. Now see the functions of plasma membrane. How the plasma membrane is helping to the cell. It permits the entry and exit of some materials in and out of the cell. It prevents movement of some other materials not required for the cell, as it acts like a selectively permeable membrane. So it is a protective guard for the cell, and it allows only some substances which are important to that cell, and it will allow some substances to go out, not all. Now let us study about cell wall. So cell wall is the outer outline of the cell. This is the outer outline of the cell. This is called as a cell wall. Cell wall is another rigid outer covering in addition to the plasma membrane found in the cell. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose. So this is very important. When we will take the plant cell, it is the composed of cellulose. Cellulose is a composition of cell wall. Cellulose is a complex substance which provides structural strength to the plant. Now functions of cell wall. Cell walls permit the cells of plants, fungi and bacteria to withstand very dilute external media without bursting. So this is hypotonic. Just now we studied what is hypotonic, hypertonic, right? The cell walls permit the cell of plants, fungi and bacteria to withstand very dilute external media without bursting. So it may be a give support with the help of hypotonic process. In such media, the cell tend to take up water by osmosis. So what will happen in this cell wall? The cell wall helps to take the water from the process of osmosis. The cell swells, building up pressure against the cell wall. The wall exerts an equal pressure against the swollen cell. So how the cell wall is help to the cell? It will help the cell by the process of osmosis to take the water, and always it will exerts the same pressure for the swelling of the cell. Because of cell wall, cells can withstand much greater changes in the surrounding medium than animal cell. So what will happen when, because of cell wall, the cell wall can withstand much greater changes, yes, in the surrounding of the medium than animal cell. Now see the process of plasmolysis. Just now we studied how the plasmolysis will be held. The water concentration is more in outside compared to the cell. That time plasmolysis we can say. When a living plant cell loses water through osmosis, there is a shrinkage or concentration of the content of the cell away from the cell wall. This phenomenon is known as a plasmolysis. So we may say that when concentration of water is less outside, that time the water will be loose by the cell, and here the water concentration will be increased and the shrinkage of cell will be happen. This phenomenon is called as a plasmolysis. Now see the structure of nucleus. This lavender color ball which is cut at one side, this is called as a nucleus. It is called the brain of the cell as it controls all the activities of the cell. It is called as a brain of the cell. Now you can see the structure over here and the label part as we seen the studied at the structure of the cell. Nuclear pores are present. It is the gateway of the nucleus which allowed some particles to go inside the nucleus. Then here chromatins are present, nucleus is present, nuclear envelope, that is outer membrane, is present. Yes. Now the next is composition of nucleus. How the nucleus made up of? The nucleus has a double layered covering called as a nuclear membrane. It is called as a nuclear membrane. The nuclear membrane has pores which allow the transfer of material from inside the nuclear to the cytoplasm. The nucleus contains chromosomes which are visible as a rod shaped structure only when the cell is about to divide. So, when the cell is about to divide, that time the size of the nucleus is increased. So, because of that we can able to observe the chromosomes. Now, so functions of chromosomes. How the chromosome is look like? It is look like this. So what are the function of chromosomes? So we can call them as a carriers of hereditary characters. Chromosomes are called as a carriers of hereditary characters. Okay, so chromosomes contain information for inheritance of features from parents to the next generation in the form of DNA. So, what is DNA? DNA is called as a deoxyribonucleic acid. We may call it as what? Deoxyribonucleic acid. Yes. So, chromosomes are composed of DNA and proteins. They will made up of DNA and protein. So, DNA is nothing but deoxyribose nucleic acid. DNA molecules contain the information necessary for constructing and organizing cells. So we may say that if a human is there, whatever his internal and external characters are there, all are present in a single chromosome. All are present in single chromosome. So that's why we are calling them as they are carriers of hereditary characters. They are carriers of hereditary characters. So functional segments of DNA are called genes. So the functional substances are called as a genes. In a non-dividing cell, the DNA is present as part of chromatin material. So, this may is present as a chromatin material in non-dividing cells. So, cells sometimes they are transferring as it is. So, they are called as a non-chromatin material. Chromatin material is visible as entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes and performs cell division. So let us tell you another thing. In a single human being, in each cell, that so in a single cell of human, uh, 46 chromosomes are present. We may say it as a 23 pairs of chromosomes. How many? 23 pairs of chromosomes. So totally 46 chromosomes are present. But when there will be a sexual process, that time only 23 pairs of chromosomes will be transferred from male body to female body, and when they will be form an egg, that time they will be comes as again 46 chromosomes, because of 23 chromosomes from male body and 23 chromosomes from female body mixed together to form as a 46 chromosomes. Now, function of nucleus. So what is the function of nucleus? The nucleus plays a central role in cellular reproduction. It is the process by which a single cell divides and forms two new cells. So one nucleus, it may enlarge, then again it will be divide, so it will become two nucleus, then that cell division will be possible. It also plays a circular part along with the environment in determining the way of cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. So this is the main function of nucleus. Now let us study about nucleoids. So this is the structure of nucleoids. This is the structure of nucleoids. So, in some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of nuclear membrane. So, students, you can observe here, this is the nucleoid. This is called as a nucleoid. Here it is written. So, it is a part of nucleus where the nuclear membrane is absent. Where the nuclear membrane is absent. So all other materials are lying as it is over here because the membrane is absent over here. Nuclear membrane is absent. So because of that the nucleus is called as a nucleoids. Such as undefined nuclear region containing only nucleic acid is called as a nucleoids. It contains only nucleic acid, that is undefined nucleic acid. That's why it is called as a nucleoid. So this is the diagram of bacteria. Here in the bacteria the nuclear membrane is absent of the nucleus. That's why we will call it as a nucleoids. Now see what is cytoplasm. Just now we studied about the cytoplasm. The cytoplasm is the fluid contained inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles performs a specific function for the cell. So what are cell organelles? You can see this is the mitochondria, Golgi bodies. So these are called as a cell organelle. Where they are present? They are present in the cytoplasm. They are present in the cytoplasm. Now see the function of cytoplasm. It helps in the exchange of material between cell organelles. It helps in exchange of materials in cell organelles. It acts as a store of vital chemicals such as amino acid, glucose, vitamins and irons, etc. So it will store which chemicals? Amino acids, glucose, vitamins and iron, etc. It is the site of certain metabolic pathways such as a glycolysis. It is the site of certain metabolic pathways such as a glycolysis. Now see the structure of endoplasmic reticulum. So this is called as a endoplasmic reticulum. The endoplasmic reticulum is a large network of a membrane bound tube and sheets. So it called as a bound sheets. Yes, these are bound tubes or we may call as a sheets. They are present in the form of sheets over here. Now see, it looks like a long tubules or round or oblong bags, or we may call as them as a vesicles. The endoplasmic reticular membrane is similar in the structure to the plasma membrane. It is also made up of lipid and protein. So these all endoplasmic reticulum all made up with the lipids and protein. Now see, in the endoplasmic reticulum there are two types. One is rough endoplasmic reticulum, another one is smooth endoplasmic reticulum. So what is the function of endoplasmic reticulum? So in rough endoplasmic reticulum looks rough under a microscope because it has particles called ribosomes attached to its surface. The ribosomes, which are present in all the active cells, are the sites of protein manufacture. So where the ribosomes are useful, they are called as the sites of manufacture of protein. The manufactured proteins are then sent to various places in the cell depending on need using endoplasmic reticulum. So wherever the protein is necessary, there it is passing with the help of endoplasmic reticulum. The soft endoplasmic reticulum helps in the manufacture of fat molecules or lipids important for cell function. So again here it is producing protein and here it is producing fat molecules or lipids. Some of these proteins and lipids help in building of cell membrane. This process is known as a membrane biogenesis. This process is known as a membrane biogenesis. Some other proteins and lipids function as enzymes and hormones. So what will happen? The main function of endoplasmic reticulum is to produce protein, lipids, and enzymes. Although the endoplasmic reticulum varies greatly in appearance in different cells, it always forms a network system. So it will be looking different and forming as a network system. One of the function of endoplasmic reticulum is to serve as a channels for the transport of material. So channels means they are called as a mediator between the transport of material between various regions of the cytoplasm or between the cytoplasm of the nucleus, especially proteins. So what is the main function? To transfer the protein. The endoplasmic reticulum also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. So it may form as a surface for the biochemical activities. In the liver cells of the group of animal called vertebrates, soft endoplasmic reticulum plays a crucial role in deoxifying many poisons and drugs. So it will help in deoxifying the poison and drugs. Now see about Golgi apparatus. So how the Golgi apparatus will be called and how the cell in where it is present. See, this is the pink color structure. This is called as a Golgi apparatus. When it will be large in size, it will look like this. The Golgi apparatus consists of a system of membrane bound vesicles arranged approximately parallel to each other in stacks called a cisterns. It called as a cisterns. These membranes often have connections with the membranes of endoplasmic reticulum and therefore constitute another portion of complex cellular membrane system. So you can see they are near always to the endoplasmic reticulum. That's why they will form as a complex cellular membrane system. Now let us study the function of Golgi bodies. The material synthesized near the end of ER is packaged and dispatched to various targets inside the and outside the cell through the Golgi bodies. So these are called as the carriers of the cell and they are transferring that proteins and other things to the other parts of the cell. Its functions include the storage, modification, and packaging of products in vesicles. In some cases, complex sugars may be made from simple sugar in the Golgi apparatus. So, complex sugar to conversion of simple sugar is made in the help of with the help of Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. They will produce the formation of lysosomes. Now see what are lysosomes. You can see the structure over in the cell, the lysosomes where they are present. Yes, in the near of mitochondria. Lysosomes are a kind of waste disposal system of the cell. It helps to keep the cell clean by digesting any foreign material as well as worn out the cell organelle. Lysosomes have membrane bounded structure whose sacs are filled with the digestive enzyme. So how the lysosomes are present? They are membrane bounded structure whose sacs are filled with the digestive enzyme. So it is called as a dustbin of the of cell. So what it will do? It will eat all the unwanted substance in the cell and it will fill the with the digestive enzyme. Now functions of lysosome. Lysosomes break foreign material entering in the cell such as a bacteria or food as well as old organelles into the small pieces. They contain powerful digestive enzymes which are made in RER. RER means rough endoplasmic reticulum, which is capable of breaking down all organic material made in a RER. During the disturbance in cellular metabolism, such as when the cell gets damaged, lysosome may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as the suicide bags of the cell. So, see, this is a very important point. Again, I am going to read. During the disturbance in cellular metabolism, such as when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are known as the suicide bags of the cell. Now, let us move towards the mitochondria. So, mitochondria are known as a powerhouse of the cell. They are known as powerhouse of the cell. Structure of mitochondria. So, mitochondria is look like this. Mitochondria have two membranes covering. The outer membrane is very porous while the inner membrane is deeply folded. These folds create a large surface area for ATP. So ATP stands for adenosine triphosphate. Adenosine triphosphate. So generating chemical reactions. So this all you are going to study in higher class. What is ATP, how it will produce. So these folds create a large surface area for ATP generating chemical reactions. Now, functions of mitochondria. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP. So, it is called as the adenosine triphosphate molecule. ATP is known as the energy currency of the cell. It is called as a energy currency of the cell. The body uses energy stored in ATP for making new chemical compounds and for mechanical work. Mitochondria have their own DNA and ribosomes. These, therefore, mitochondria are able to make some of their own proteins. So, this is also important. Mitochondria contain its own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. Now, let us move towards plastids. So, plastids are present only in plant cell. Where it is present? It present in only plant cell. There are three types of plastid. So, one is colored plastid, white or colorless plastid, and another one is pigment chlorophyll plastid. So, you can see the structure over here. So, structures of plastids. The internal organization of the plastid consists of numerous membranes layers embedded in material called a stroma. So, here it is, you can see the stroma, chloroplast, and again all the substance over here. This is the called as a stroma. The inner back structure is called as a stroma. Now see, plastids are have their own DNA and ribosomes like mitochondria and similar to its structure. So why plastids are present? What are their work? Let us see. Chloroplasts are important for photosynthesis in plants. Chloroplasts also contain various yellow and orange pigments in addition to the chlorophyll. So leucoplasts are primarily organelle in which materials such as starch, oils and proteins granules are stored. So, not only they will involve in the process of photosynthesis, but they can store the starch, oils and protein granules in them. Now see about the vacuole. You can see the vacuole. Here the vacuole is very big. In some cells the vacuoles are small, or in some cells they are absent. Vacuoles are storage sacs for solid or liquid contents. They are small sized in animal cell while plant cell have very large vacuoles. So in the animal cell they are very small and in the plant they are large. So functions of the vacuole. The central vacuole of some plant cell may occupy 50 to 90 of the cell volume. So They may be contain 50 to 90% of the cell volume. The plant cell vacuoles are full of cell sap and provides turgidity or rigidity. Rigidity means stiffness to the cell. Many important substances in the life of the cell are stored in vacuoles, which include amino acid, sugar, various organic acids, and some proteins. So, many important substances in the life of the plant cell are stored in vacuoles, which include amino acids, sugar, various organic acids, and some proteins. So, these are the materials which are important up to the birth of the cell to the death. That's why they all are present in the vacuoles. In a single celled organisms like amoeba, the food vacuoles contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important role in expelling excess water and some waste from the cell. Thank you.